These sensitive strategies can be used to evaluate the inhibition efficacy of chemical inhibitors and in a combination with CRISPR-Cas9 gene deletion to investigate the drug targets.
Instead of using an endpoint assay, parasite growth can be monitored over time to allow calculation of parasite doubling time using a sensitive reporter protein.
This assay can potentially be scaled up in 384 or 1,536 well microplates for the testing of multiple compounds or for the screening of libraries in a high-throughput manner.
This strategy can be modified to quantify the growth of other intracellular microbial pathogens and their responses to the drug of interest.
To perform a luciferase-based Toxoplasma growth assay, begin by carefully aspirating the medium from pre-seeded 96 well microplate cultures of human foreskin fibroblasts and inoculate 150 microliters of parasite resuspension into the wells in a 3-column, 5-row format. After a 4-hour incubation in the cell culture incubator, carefully aspirate the medium from each well to remove the non-invaded parasites and fill the wells in each but the first row with room temperature phenol red-free medium. Then add 100 microliters of a 12.5 micromolar luciferase substrate solution in PBS into each well of the top row and incubate the microplates for 10 minutes at room temperature to allow full cell lysis. At the end of the incubation, measure the luciferase activity on a microplate reader. Each reading represents the initial number of invaded parasites at 4 hours post-infection. Repeat the analysis every 24 hours for the next 4 days without changing the medium.